Meanwhile, researchers say type O blood appears to offer people some protection against the coronavirus. Two studies published in Blood Advances suggest people with type O may have a lower risk of coronavirus infection than those who have other blood types.
You're fascinated by the edge of discovery. You're in the right place. This channel explores mind-blowing breakthroughs, hidden truths, and science's deepest puzzles, all backed by the latest research and findings. We're not just scratching the surface. We're going deep into the why and how of the world's greatest mysteries. Whether it's ancient genetics, biomedical breakthroughs, or the evolutionary secrets locked in your DNA, our mission is to deliver the facts, decode the science, and share the human story behind each breakthrough. Today's deep dive is one that touches every human being's blood type, and not just any type. The elusive, fascinating, and life-saving type O blood. What makes it so special? And why is it particularly mysterious in the Americas? In 2025, scientists finally cracked the code. Let's dive in. Type O blood is more than a classification. It's a key that unlocks mysteries in emergency medicine, genetics, and human history. Its biological value is enormous, especially when seconds count. In trauma situations, doctors don't always have time to type a patient's blood. That's where type O negative becomes critical. It can be transfused into anyone, regardless of their blood type, making it the default option when there's no room for error. This is why every emergency room, military base, and ambulance aims to stock it. Type O positive also plays a crucial role. It's the most common blood type worldwide, especially in places like the Americas and parts of Europe. While not universally accepted like O negative, it can be transfused into any patient with a positive Rh factor, making it incredibly useful in large-scale emergencies where blood demand spikes. But what really turns heads is type O's strange global pattern. Scientists noticed something unusual when mapping blood types across continents. In certain Native American tribes, especially in South and Central America, virtually 100% of individuals had type O blood. That's not just statistically rare, it's biologically astonishing. In evolutionary terms, genetic diversity is the norm. Over time, interbreeding and migration tend to introduce variation. So even in relatively isolated populations, we'd expect to find a mix of A, B, and O alleles. But with some indigenous American groups, those other types are practically non-existent. This led scientists to wonder, how could such a narrow genetic trait persist in entire populations? For decades, researchers theorized, some believed it was a founder effect, that the first small groups to migrate into the Americas from Siberia during the Ice Age happened to carry only the O allele. Others pointed to natural selection, suggesting type O may have offered a survival advantage in environments plagued by specific pathogens. Then, in 2025, a breakthrough arrived. Genomic studies comparing ancient DNA from mummified remains, preserved humans, and early American settlers revealed a strong pattern. The people who first crossed the Bering Land Bridge roughly 15,000 years ago came from a genetically narrow group. These early humans likely underwent a genetic bottleneck, a period when their numbers were so low that only a limited gene pool survived. And in that pool, the O allele dominated. What's more, it turns out that this allele conferred a modest advantage in terms of resistance to certain infectious diseases, such as malaria and tuberculosis. While not completely immune, type O carriers were slightly more likely to survive outbreaks, allowing their bloodline to flourish while others diminished. Over generations, this created a sort of genetic echo, type O blood embedded deeply into the fabric of American indigenous DNA. Even today, that ancient fingerprint persists. Across Latin America, type O remains disproportionately high, even in urban centers with diverse populations. It's a lingering clue from humanity's ancient migration story, one told not in bones or artifacts, but in the living tissue of blood. Understanding Type O's role isn't just an academic pursuit. In a world increasingly reliant on blood transfusions for surgeries, childbirth, and emergencies, knowing where and how this blood type is distributed helps global health systems plan better. It shapes blood donation campaigns, informs medical research, and even offers insights into how our ancestors adapted to a changing world. So, the next time you see a call for blood donations, especially for Type O, remember, you're not just giving blood. You're continuing a legacy that has flowed through humanity for thousands of years, a lifeline connecting past, present, and future. The founder effect has long been a cornerstone theory for explaining the unusual dominance of Type O blood among indigenous peoples of the Americas. According to this idea, when a small group of early humans migrated from Siberia into North America across the Bering Land Bridge around 15,000 to 20,000 years ago, they carried with them only a limited set of genetic traits. If this migrating population was largely composed of individuals with Type O blood, then, through generations of reproduction within an isolated gene pool, the O allele could have become predominant by default. This kind of genetic bottleneck isn't unusual in evolutionary history, but as researchers dug into this theory, it started to show cracks. While the founder effect explains how a single trait could become common in a small, closed population, it doesn't account for the near-total absence of the A and B alleles. Over the span of thousands of years, you would expect genetic mutations to reintroduce these alleles. Even modest interbreeding with other human groups would normally result in more blood type diversity. But in many Native American tribes, especially those isolated in the Amazon or high Andes, A and B types remain vanishingly rare, sometimes entirely absent. So, if not just genetic drift, then what? This opened the door to another possibility: natural selection through environmental adaptation. Could the environment itself have favored individuals with Type O blood? Researchers began looking at specific traits tied to the O allele. One of the most compelling lines of investigation centered on disease resistance. Type O individuals, it turns out, tend to fare better against certain diseases. They're less likely to suffer from severe malaria, for instance, a disease that, while not historically widespread in North America, may have been a factor in parts of Central and South America. Even a slight survival advantage in these regions would, over centuries, lead to more Type O individuals passing on their genes. Another fascinating angle came from studying digestion and metabolism. People with Type O blood typically produce more stomach acid and possess a robust gut biome optimized for protein digestion. This would have been advantageous in early human diets that relied heavily on hunted meat and gathered food with fewer carbohydrates and grains. In environments where agriculture hadn't yet taken root, this digestive edge could have significantly improved survival, nutrient absorption, and resistance to pathogens in food. Still, these remained educated guesses until 2025. Year, an international team of geneticists and anthropologists conducted an unprecedented genomic study. By analyzing both modern and ancient DNA samples across the Americas, they were able to construct a high-resolution map of blood type distribution over time. What they discovered was groundbreaking. The persistence of Type O wasn't just due to the founder effect. It had been actively preserved by selective pressure. Their data showed that individuals with the O allele marginally higher survival and reproduction rates in certain environmental contexts, especially in regions with high exposure to zoonotic pathogens, where immunity and digestive efficiency played key roles. Over time, natural selection reinforced the dominance of the O allele in these populations, effectively crowding out A and B. In other words, evolution had a hand in preserving what the founder effect had started. The combination of a narrow genetic starting point and selective advantages turned type O into not just a common trait, but a defining one for entire populations. Today, this discovery reshapes how we understand human migration, adaptation, and survival. Blood, once thought to be a simple hereditary marker, now reads like a storybook, one that tells of isolation, resilience, deep biological memory of the environments that shaped us. In 2025, a groundbreaking genetic study redefined what we thought we knew about human blood types. Conducted jointly by Harvard University, UNAM in Mexico, and the Max Planck Institute in Germany, the project leveraged next-generation sequencing to analyze thousands of DNA samples from indigenous populations across the Americas. What they found was astonishing. A previously undiscovered sub-allele of the type O gene, now called O', was present almost exclusively in Native American genomes. This wasn't just another variant. O showed unusual genetic stability, meaning it had resisted mutation for thousands of years. Compared to other blood type alleles, which tend to evolve and shift over time, O had remained virtually unchanged since its earliest appearances. What shocked researchers even more was its apparent age. O wasn't just another version of O, it might actually be the oldest of all blood types. This discovery upended decades of medical and evolutionary thinking. For years, the dominant theory held that type O was a stripped-down, simplified mutation of the more complex A and B types. But with O predating them all, a radical new hypothesis emerged. Type O may have been the original human blood type, and A and B might actually be the mutations, adaptations that developed later in response to environmental pressures and migration patterns.